Okay, sorry about the audio issue. I'll just quickly go through the slides again to recap. I was saying that cancer is still one of the leading causes, and only very small amount of cancer patients participated in cancer therapy trials. So it makes it very difficult to recruit patients and maintain the cost for clinical trials, especially for later phase trials. Now, when the oncology treatment paradigm shift from targeting same tumor histology to more precision medicine, the patient population is more refined, which makes it even harder to find participants. Under that context, innovative clinical trial designs are urgently needed for better efficiency while still maintaining the trial quality. Bayesian methods may help mitigate this challenge. In early 2024, the US FDA launched a new center specifically for clinical trial innovation, and exploring Bayesian statistical methods is one of the first 3 innovative design demonstration programs. In Bayesian statistics, some key information we need to understand is that the unknown parameter of interest, such as a risk ratio, is a random variable with a true distribution among a population instead of one fixed true value as what we usually consider in frequentist statistics. In the analysis, we need to first specify a prior distribution based on existing knowledge, and after collecting new data, we will then use the data to update the distribution, which is referred to as the posterior distribution. With the classic frequentist methods, we would specify key parameters such as estimated effect size, type 1 error, power to calculate the target sample size with a formula. But with Bayesian methods, we would incorporate the prior information and conduct simulation studies under different scenarios and assumptions to estimate the target sample size, as well as the false positive and false negative rate under each scenario. Sorry. So in Bayesian methods, the estimated sample size may be a range, and we may gain efficiency in sample size when the prior and other assumptions made are appropriate. Prior distribution can be based on many sources, including natural history of a disease, results from prior studies, data from other arms of the concurrent study, according to how applicable the existing information is. The prior can be set as strongly, weakly, or even not at all informative. And intuitively, the more informative the prior is, the more it is likely to reduce the sample size. Bayesian methods also allows for more frequent interim analysis. In classic frequentist test, to avoid inflating the type 1 error, we need to be cautious on how many times we look at the data, so the number of interim analysis is usually limited. But in Bayesian statistics, type 1 error does not apply in the same way. Since the true effect is considered as a distribution, we can define a treatment being effective when the majority of the posterior distribution of the measure falls within the range that indicates a clinical meaningful effect. And the majority and clinical meaning, what is majority and what is clinical meaningful effect need to be pre-specified. For example, if we define the majority as greater than 80% and a risk ratio of less than or equal to 0.6 as clinically meaningful effect, then the treatment is effective when we see most of the posterior distribution, like more than 80% of the posterior distribution falls under risk ratio of 0.06. And under this paradigm, the classic type 1 and type 2 error concepts do not apply, and the false positive and false negative rates are usually summarized by simulation study results. So in that circumstance, we can plan for multiple interim analysis as desired, and that would give us more chances to detect futility or efficacy to stop the trial early or making adjustment along the way. And this could help reduce actual sample size and shorten trial duration as well. Despite these advantages, Bayesian methods may still be underutilized, especially in later phase trials. A review based on clinicaltrial.gov records showed that only 225 Bayesian oncology trials were found from 1990 to 2020. And most of them were phase 1 or 2 studies, with cancer institutions and hospitals being the main funding sources. So why is that? Here are some potential obstacles and concerns of using Bayesian methods. First, stakeholders such as regulatory agencies and physicians lack familiarity to Bayesian methods. Most of the literature on Bayesian methods are very technical and hard to understand for non-biostatisticians, and very few reviews have summarized Bayesian trials that are has conducted in the past, and they were based only 1 or 2 databases. And to address this concern, to address this gap, we plan to conduct a scoping review first to understand and summarize how Bayesian methods were used in the past, with a focus of later phase oncology trials. Second, if Bayesian methods can actually lead to better efficiency in practice is not evaluated. Inappropriate assumptions made in simulation, as well as extensive communication and additional coordination needed due to the complexity in design, may override the potential efficiency Bayesian methods are expected to offer. To evaluate this, we propose to compare the actual sample size and trial duration between completed Bayesian and frequentist trials and provide information on how Bayesian trials performed in practice. And lastly, the impact of increased trial design complexity on the quality and validity is unknown. And to understand more of these, we will evaluate the risk of bias and aggregated false positive rate of Bayesian trials compared to frequentist trials in cancer therapy development. First, aim 1. We are going to map out where and how Bayesian trials were used in cancer therapy trials. We're going to conduct a scoping review searching for Bayesian trials that evaluated efficacy of cancer therapies. We will search on both literature databases and also clinical trial registries, and 6 databases in total. We are going to include the Bayesian trials that are phase 2, seamless phase 2 and 3, and also phase 3, investigating cancer therapies, including drugs, biologics, radiotherapies, etc., but exclude surgery or medical device. Evaluating treatments efficacy has to be at least one of the primary objectives of the trial. And we won't set any restrictions on the year of the trial or the publication. Data collection will be focusing on capturing data, trial features, and also Bayesian methods information. Trial features may include administrative information, trial design, innovative features, results, and regulatory experiences. And Bayesian methods, we will be looking at the function of Bayesian methods used, the prior, the statistical model, and also the results. One of the major challenges in this aim is that the scope of the review is hard to estimate at this point. But if there are too many or too few trials, we will then adjust by restricting the trial initiation year, or expanding the project to other disease areas. By doing the scoping review, besides enhancing the scientific community's awareness and understanding, we also aim to identify some factors that are highly likely to be associated with the use of Bayesian methods, and to use that to inform the analysis in aim 2 and aim 3. For aim 2 and 3, here are some common setups. The key elements in study design. Instead of having human participants as the study subject, we're using completed clinical trial as the subject. And the exposed group are defined as trials using Bayesian methods for trial monitoring or primary efficacy analysis. And the control group will be defined as trials use frequentist methods for trial monitoring and primary efficacy analysis. And the outcome includes trial efficiency measured by sample size and trial duration, and also trial quality measured by risk bias and aggregated false positive rate. We expect to find some master protocol trials that use Bayesian methods. These trials usually have more than one sub-studies under a master protocol, and there may or may not be information borrowing across sub-studies, but they're likely to be correlated. So to address this, we are going to construct the included trials with this clustered structure. Each sub-study is treated as an individual trial, and the master protocol trial will be treated as a cluster. For example, if a master protocol has n sub-studies under it, then the cluster size will be n. And if a trial is not a master protocol, then it will be its own individual trial with cluster size equals to 1. Large platform trials with no explicit start or end date will be really hard to include in the analysis, so we will exclude those trials. To find control frequentist trial, there are 3 main sources. For each Bayesian trials we identified, we will look for the systematic reviews that included the Bayesian trial and extract the list of all trials included in this systematic review as the source 1. And we will also collect the reference list and citation list of the Bayesian trial. And from those lists, we will then identify up to 4 frequentist trials as the control group by matching on disease, phase, endpoints, and randomization. If the Bayesian trial has no publication available, then we will go to the trial registration records where we find the Bayesian trial to identify the control frequentist trials. And if that is also not available, then we will have to exclude the Bayesian trial as well. From a pilot look on MBase, we find 25 Bayesian trials out of 234 records. So a rough estimate, we will have 50 to 100 Bayesian trials and 100 to 500 trials in total. After identify all the Bayesian trials and their controls, we're going to first compare the trial efficiency in aim 2. In part 1, measuring efficiency by sample size, we'll be looking at estimated and actual sample size, as well as the ratio between those two. First, we'll plot the estimate sample size and compare 2 groups visually. And for the primary analysis in this part, we're going to test the hypothesis whether Bayesian trials had less actual sample size than frequentist trials using mixed effect linear regression model. And to prevent data being skewed due to the large trials, we're going to transfer the sample size to the log scale. And we're also going to control for key factors that will affect the sample size, such as being in adaptive design trial, number of arms, expected sample size, type 1 error, power, etc. And for the secondary analysis, we're going to look at the ratio of actual sample size and estimated max sample size. Instead of doing hypothesis testing, here we're going to estimate the ratio and its 95% confidence interval for each group. And for Bayesian trials specifically, we want to see what's the proportion of the trials that actually reached or exceeded the estimated maximum sample size and explore the potential factors that are associated with it. In part 2, we're measuring efficiency with trial duration. Ideally, we want to measure the whole process, including the initial planning stage, but it is unlikely to know when the project initiated. So we're going to count the start of the trial from the date of the first protocol. The study completion information will be defined as the time when the last patient's final measurements are collected. And this information is normally available in publication and also the clinical trial registration platform. After trial completion, we will also count the analysis and also the publication time together as the analysis and dissemination time. We're going to look at the trial operation, analysis, and dissemination separately, and also adding them together as the total trial duration. For all those 3 outcomes, we will estimate the mean difference in months and calculate its 95% confidence interval. For the trials that results are not published anywhere, we will only look at the trial operation time. In this aim, the biggest concern is the protocol, first version of protocol or later may not be available for trials, especially for the trials that conducted decades ago. So for this, we're going to utilize publications and trial registration platform, maybe also the FDA database to collect key information. And if the outcomes that we are interested in aim 2 are missing, then we will have to exclude the trial. After evaluating the efficiency, we also want to make sure the complexity does not affect the trial quality. First, we're going to measure the trial quality by risk of bias using the risk of bias measurement tool for randomized and non-randomized trial, because the outcome will be a ordered categorical variable. We're going to use ordered logistic regression with robust variance estimator to compare the 2 groups. And because the tool will be different among randomized and non-randomized trials, we're going to stratify the analysis by randomization. For this one, because we want to see if Bayesian trials would affect the quality, so we're setting the hypothesis as non-inferiority hypothesis to see if the risk of bias is higher. The second part is to measure the quality using aggregated false positive rate. For each phase 2 trial with positive results, we're going to look for its subsequent phase 3 trial and see if the results between those 2 are concordant. If yes, then we would conclude that the phase 2 positive results is a true positive result, and if no, we will conclude that the phase 2 trials is a false positive results. By that, we will then calculate the aggregated false positive rate by dividing the number of false positive phase 2 trials by the total number of phase 2 trials that showed positive results. And having informative or non-informative prior may affect the type 1 error or false positive rate. So in this analysis, we're going to stratify by the prior type. This is also going to be a non-inferiority hypothesis testing, and we're using mixed effect logistic regression to do the analysis. The major limitation and in this aim is the subsequent phase 3 trial may not be available. Under that circumstance, we will summarize and report the proportion of phase 2 trials with positive results that subsequent phase 3 trials cannot be found in each group, and see if it's a big issue and evaluate from there. To recap, this is the overall picture. We're looking at later phase Bayesian trials evaluating efficacy of cancer therapies. We're going to give it a scoping review to map out the use of the Bayesian methods, match each Bayesian trial with frequentist trials, and then compare the efficiency and quality between those two groups. Through this project, we hope to enhance the scientific community's awareness and understanding of Bayesian methods in practice, and also to provide evidence on whether Bayesian methods could actually bring efficiency while maintaining trial quality in cancer therapy development. And that's all. Thank you. Okay. Open to questions now. I can. Hello.
I can. I could tell that Shariza spoke, but I did not hear the question.
Well, that was my question, but we were trying to figure out if she could hear you guys online, and she can.
Yes, I can. And I can also repeat out the question to the room. I will need to work on the acknowledgement.
I just want to make sure that I understood it all. It was a very nice presentation and put together well. But what I was wondering is, so you're going to get your population of Bayesian trials is going to be identified in aim 1, and then you'll use those trials to match to other trials from to create a control group from the database of clinicaltrials.gov?
Can you hear me? Yes. So yes, clinicaltrial.gov is one of the sources that I'm going to look for controls. We'll also look for controls through systematic review and the citation list. For example, if I'm finding, for each Bayesian trials, I will trace back to the systematic reviews that included this Bayesian trials, and then look through the list of trials that systematic review has included, and see if there are frequentist trials that studies same disease in same phase with the same randomization scheme, and pick them as a control group. I'm also going to look at the publications that referred by the Bayesian trials, or the publications that cited the Bayesian trials, to look for frequentist trials that's similar to the Bayesian trial identified.
So matched controls, is that what you're saying?
Yes. So that's one of the concerns. That's why I didn't really put drug over here. I'm only matching disease. And with drug, especially for false positive rate, that we understand that the effect size might be a big issue if the trial doesn't work. If the drug doesn't work, then it doesn't work. To address for that, instead of matching by drug, because it's going to be really hard, we're going to put the key variables, like if the expected sample size, type 1 error and power, into the model as one of the factors that to control for drug effect. Thank you.
I think everyone's kind of asking about the controls, and I think it's a way kind of it. So my question is, if you're looking through the references, what happens if one of the frequent trials is kind of what informs the prevalence information for the Bayesian trial to occur? So let's say like the control, like the frequentist trial, let's say there wasn't a known base rate of whatever genetic mutation. The frequentist trial kind of helped inform that, and then that gave kind of the posterior information for the Bayesian trial to even occur. Doesn't that kind of set it up for the Bayesian trial to perform better?
Mm-hmm.
Information from a control trial.
Mm-hmm. That's a really good point. I have not thought of that. That means that the frequentist and Bayesian trials may not be independent of each other.
Yeah.
But as far as I'm thinking about it now, because in the false positive analysis, we're going to stratify the analysis by prior knowledge, and by stratifying the analysis by prior type, then this will be the situation that you're mentioning will be having a informative prior. So I guess that could be one of the way to address this the coordination, the relation, by putting prior as informative prior as one of the variable that affect the results. And in actual practicing context, the whole point of having Bayesian trial is to borrow information from previous trials, and that's kind of one of the factors where we are trying to evaluate. I think if we are controlling on that factor, then this whole project will not make sense. Does that make sense?
Yeah, I think you're in the right realm, kind of like think for it, account for it.
Yeah, I think that's a really good question. I will note that down and then think about it more, but I think having, if I put the types of prior as one of the factors in the model, it might be able to address for the, and then also because I'm using mixed effect model, this model will help me address for any coordination or correlation between observations. So hopefully that could also capture the correlation between the groups.
Yes, you'll have to have a more or less precise definition of informative and non-informative, and what is the dividing line.
Okay. Yes. Up to 4. I doubt there will be that many. I would be very happy if I can find one, but if there are a lot, then it will help me increase the sample size of the analysis.
I don't understand exactly what you mean by the false positive rate. You're going to calculate that theoretically, not was the outcome of the trial incorrect because what we've learned in practice, that the drug really works even though the trial said it was negative? You mean it's a calculated false positive rate theoretically, right?
So under this context, I'm going to treat the phase 3 results as sort of the gold standard. If
Oh,
I assume that if a phase 2 trial has a positive results, then there will be a follow-up phase 3 trial, either before the approval of the drug or after the approval of the drug. And I'm going to use that results as the gold standard. And if the phase 3 is also positive, then I will conclude that the phase 2 positive is a true positive. And if the phase 3 confirmative trial concluded negative results, then we will say that the phase 2 positive results is a false positive.
right. Well, then for the phase 3 trial, you're not going to compute false positive. It's only for the phase 2.
Yes, only for phase 2.
Right. Well, then you may find the phase 3 trial is not the same as the phase 2 trial because of what they learned in the phase 2 trial. So they may have different eligibility. They may have different doses, schedule. That's why we do phase 2 trials. So it may not be in all cases just a smooth transition from phase 2 to phase 3.
I see. So I see. Because sometimes the endpoint will be different and those will be a little different.
Well, in phase 2, I mean, in oncology, very often we use the response rate.
Mhm.
And then phase 3 would be progression-free survival or overall survival. And that is true. Endpoint, eligibility, dosage, those could all change. In the diseases I am familiar with, in other diseases, perhaps infectious disease, it will be a smooth transition.
Mhm. To this aim, what I'm thinking right now is to look at the implication. So if the phase 3 is evaluating different population or different endpoint, but the result is that the drug is not effective in this population, that
Yeah, that's good. No, I think that's a good explanation. Regardless of how it's done, was the phase 2 predictive or was it not? That makes sense.
Mhm. Yes. I can repeat the question. Mhm.
I think I have some quibbles with merging in false positives and positive quality. I don't think that's a reflection of quality, it's a reflection of all these things.
True.
So I wonder whether we can still measure it, we have it, but it may not be neat enough, but you capture that part somewhere.
Mhm.
Please offer quality. We know that the false phase 2 and phase 3 trials necessary. The other thought, just in response to what Shirley was saying earlier, I think what you might consider about the treatment is for informing. I wonder you might consider a sensitivity analysis in any trials that you think potentially or actually could have informed the treatment this trial.
Okay.
And then compare the rest and see whether the results
Okay.
informing trial.
Okay. That's a really good, and I'm gonna note that down. Oh, Stefan.
Hey, thanks. So I couldn't hear anything that was said in the room. So if this is redundant, just let me know. I was just questioning whether some of those comparisons might be confounded just by the indication for doing a trial. So those Bayesian trials, it seems to me, they are different and sometimes quite different from the frequentist trials. And sometimes that's because of a specific indication. So for example, it's a very, very rare condition or what have you. So that's, I guess my question is, have you considered confounding by indication, confounding by condition, by disease severity, something like that?
So in the matching strategy, I am going to match the frequentist and Bayesian trials by the disease. I should probably phrase this as more accurately as indication, but I am at this point not sure if I can't find exact indication matches the severity and also the disease and potentially the biomarkers that define the subgroups. So I will need to explore more in terms of how stringent I want the matching be in terms of the indication and disease. But regarding to whether or not the disease prevalence part, I'm also thinking of putting whether or not the disease is a rare disease as a binary indicator to the model, because that does affect the sample size calculation and also trial duration.
Yeah, good. I would almost think like it's something like a qualitative overview. Sometimes people would especially when they have a Bayesian design, they would say, we designed this trial as a Bayesian trial because, and then you get some idea why that design was chosen relative to a frequentist design to kind of get at that, because we don't want an unfair comparison. So we want to try and understand when these designs are used, I think. So I think that might be informative.
Okay, sounds good. I will put that on the goal or the aim of using Bayesian trials as one of the data collection point.
Yeah, and one other comment, you talked of adaptive designs and that use Bayes. I think I-SPY 2 comes into mind as a basket trial. That is Bayesian, yes, although how it's used remains a mystery to most. But the thing is, different drugs are rotating through. And they have their time in the limelight, and then they move out. And I don't know how you're going to compare. Well, you need to be careful. Are you going to take one drug in that rotation, say if it was I-SPY 2, and then compare it to a phase 3 if it ever came to phase 3? Or are you going to try to take each drug as it came through and compare it? Because I don't, that's quite a mix. I think that's a much too ambitious undertaking in what we understand from I-SPY 2, because so much of it is considered proprietary. And I don't know even, is it an informative or not informative prior? And they're doing all kinds of things of going back in time and correcting for how long ago the control group was. I wouldn't, I guess if it's I-SPY 2 that you're thinking of, or any of these basket trials that use Bayes, they're very good for, at least in oncology. It's very important to have them, but I don't think it fits in what you are doing, or that it will be easy for you to do what you want to do. I would just stick with the more conventional trials.
Okay. Yeah, that's I-SPY 2 is one of the example that I thought of that made me decide to exclude those huge platform trials because they're way too complicated. And it's, as you said, it's very ambitious to try to figure out each drugs are coming in and out, figure out the time and the follow-up trials. So under those circumstance with there's no is like running forever trials, no defined end or start date, I will exclude those trials, since it is, I think it's more, those trials have their own chemistry or structure that is not just Bayesian.
It's a different beast than what we normally think of as phase 1, phase 2, phase 3. So
Right.
I would leave it out, but not because they're not an important innovation in clinical trials, but because it doesn't fit your spectrum here.
Okay, thank you. Thank you for confirming that, because I was also debating how to deal with it, but I think having your confirmation is good.
Right.
Right,
So, I was just wondering if you would, again, make sure that you're, I think it's gonna be.
Thank you.
Where are the points where you-
Okay, so the question in the room is, because this is a practice round for the proposal seminar and this presentation is actually shorter, I will need to put in more information. Do you have any recommendations or ideas where I could, or I need to put in more information?
I think in the background section, when you first introduced the problem, I wasn't convinced that all of your aims work together. So you could add an overarching question and bring in some either guidance documents that it could pertain to, some sort of pushes within the FDA, even within other agencies across the world as well, because I think your study question is very pertinent, but not just to FDA, but to all the regulatory. Something along those lines might help. And then at the end, with the impact and of course how you did touch on how you're going to tackle some of those challenges, but I think you could delve in a little bit more, especially with the heterogeneity you're going to face. That is going to be a huge hurdle to overcome. Are you going to plan any subgroup analyses? How are you going to handle that for each of the aims? I think that'll be critical too. You want to head off some of those questions within the same.
Okay. Potential limitations.
Yeah, this doesn't relate to a future presentation, but one thing I had thought of, it is important to distinguish between informative and non-informative priors, but I think most of these trials are going to have non-informative priors because they're going to go before FDA, and FDA was they're not very receptive often to Bayes and this subjectivity of choosing a prior. So non-informative is also conservative. So I'm not sure you're going to find many informative priors in the literature, but good luck with it if you do, and if you don't, then you just don't include that. And it may be good because of most of them are non-informative. It makes for a better comparison.
Okay, sounds good. And also, one of the goals in aim 1 is to figure out how much those things are and how they actually worked in the past. And if most of the prior is non-informative, I think that would be a really good information point in aim 1.
Yeah.
Thank you. Yes.
For aims, you may have to repeat the question, but aims 2 and 3, you are restricting the studies that are complete.
Yes.
What might be the implications of that? Might there be differences between the data and frequency So, think about what, what
So the question in the room is, in aim 2 and 3, I'm only including the completed trials, and what could be the implication of that, and may need to think about how to, whether or not Bayesian has the effect on whether trial can complete or terminate or something. So with that point, I'm going to actually collect information, whether or not it's terminated, completed, or stopped early in aim 1, and summarize that in aim 1, the scoping review, to map out the completion rate, not compare, but just to talk about the completion rate of Bayesian trials. Right. I guess there's no formal comparison between Bayesian and frequentist trial, which is probably one of my limitations. Yeah.
Any other questions? You have questions you want to ask us?
Question. So I think overall, the part that I am most concerned of is also how to find the good match, as most of the question has been focused on, because it's very easy to be confounded by other factors. So I would appreciate if there's any thoughts on the matching factors. I think Stefan mentioned the indication specifically. If there is any other thoughts on what factors that I may left out. Yes.
I feel like, you know, the kind of scoping you're doing as part of aim 1 is going to be informative.
Right. Right.
So is like your protocol enclosed for like aim 2 flexible? Like do you think it would change things based on, or are you gonna leave it like based on what you learned from gathering information in aim 1?
Yeah, that's my goal, to look at. But one of the issue is, as we the comments in the room is whether or not the results of aim 1 will inform the aim 2 and 3 design in that sense. I think yes, I will definitely use the information that I found in aim 1 to help design aim 2 and aim 3. But one of the issue that I am thinking of now is because in aim 1 there's no comparison, so I wouldn't know if for example, the completion rate, whether or not it's higher or lower, or if it's associated with the use of Bayesian trials. So that might be one of the pitfalls. At this stage, I'm trying to conceptualize look at what factors might be important confounding.
Well, what you want to look at before you start this is what do you want your final sample size to be with all of the matching? You don't want to go through all of this work and do the matching as best as you can and wind up, say, with a sample size of 30. That might be too small to make inferences. Do you want it to be 100? Well, if it's going to be 100, you've got to broaden your matching characteristics. That's going to be a trade-off, and you may have more insight, and your advisors more insight on that when you start. But keep in mind, you don't want to go through all this work and find there's only 25, 30 matches that you can analyze.
Okay, sounds good. Like go to more
Strategy.
databases? Potentially, or we're thinking of expanding the databases that we're going to look for frequentist trials to see if we can find more matches. And also another thought in head, I haven't talked to my advisor team yet, but another thought I have is if in any case the matching fell through, then for all the Bayesian trials, most of them, as far as I know, that they would also report the summarized type 1, like the statistical operating characteristics. Then I could use those statistical operating characteristics to calculate the frequentist as estimated sample size and compare those estimate sample size to the actual Bayesian trial sample size. But then one of the concerns with this mitigation strategy is then I wouldn't see the execution of the frequentist trial and it go back to simulation again. So then I need to figure out a way to incorporating the execution part into that comparison with that strategy. So I definitely need to think more about it. Maybe I can have a call with you, Jay, to talk about simulations more, but that's one of the thoughts I have.
I see Chen, who is one of your advisors. He would be very knowledgeable in these matters as well.
Okay, sounds good.
Last question?
Last questions? Okay, thank you all. All very good advices, and I will go back and think about all the points that was raised today. Very helpful. Thank you.
Okay, good luck.
Thank you. Thanks.